Hello, everyone, and welcome to ALS M&D Connect 2021. My name is Kathy Cummings. I'm the Executive Director of the International Alliance of ALS M&D Associations, and I apologize for that little pause. I have my grand cat here with me, and she decided to step on my computer just as I was starting to speak, of course. I would like to welcome you to this fantastic panel of international researchers who are going to bring us the latest and greatest updates and what's happening in the research field. And I really want to thank our sponsors who make all of this possible. We had some fantastic partner sponsors for all of our meetings that we've held over the last month. We have Biogen, MT Pharma, Amylyx, UCB, Apellis, and our foundational sponsor, Cytokinetics, that have enabled us to do this. We also have some sponsors specific to ALS M&D Connect. We have Brainstorm, Biogen, and Amylyx, and we really appreciate their support. We couldn't do what we do without the support of our industry partners. Today, it's my pleasure to welcome Dr. Felipe Ocampo to kick things off for ALS M&D Connect. Felipe is a medical surgeon who currently resides in California with his husband and 5 dogs. And I'll let him tell his own story about his diagnosis with ALS M&D in February of 2021. And he's a huge supporter of research. So without further ado, let's hear from Dr. Felipe Ocampo.
Hello everyone, welcome to this ALS M&D Connect. My name is Felipe Ocampo. I am a medical surgeon and I have been living with ALS M&D for 9 months. This is an honor to speak to you today. Thank you for having me. My journey started when I started losing my strength on one of my hands. I thought maybe my body was tired because I would exercise every day and I would lift weight on my daily basis. I thought maybe it was a strain. Time keeps passing and I was not getting better. From my medical background, I knew this was not normal and something else was going on with my body. I visited my doctor and explained my issues and how I was losing strength and had fasciculation on my arms. She said I would be okay and that it was maybe from a muscle strain. After that visit, I was not getting better and I started to really worry and needed to find out different tests. I was diagnosed with ALS M&D in February of 2021. So my first reaction was go to my laptop and search the internet about ALS M&D, as I did not know the depth of this disease. The first thing I found was that my life is going to be over in 2 or 5 years. Really scary information, as I felt my life was over. However, I only had 2 options: waiting to die or start fighting against it. I joined some ALS MND association and I found out that this disease is different in everyone. Besides, it is possible to live more than 5 years. That's why research is really important. We can learn more about this disease and how to treat it. There is not too much trustworthy information out there or accessible sometimes. Any new data is going to bring benefits to people and science. We want a future with not ALS MND. Thank you to everybody who invests time on research.
Thank you so much, Felipe, for that reminder of why research is so important. It's now my pleasure to turn the program over to Dr. Nortina Shahrazalia from Malaysia. Nortina is a graduate of the University of Nottingham Medical School in 1997. She completed her general medical training, specialist neurology, and doctorate degree in the UK before returning to Malaysia in 2009. As part of her specialist neurology and neurophysiology training, she worked at a tertiary neurology center, including Queen's Medical Center, Nottingham, and the National Hospital for Neurology and Neurosurgery, Queen Square, London. She is currently a consultant neurologist at the University of Malaya Medical Center, as well as a professor of neurology at the University of Malaya. She has a subspecialty clinical and research interest in the field of neuromuscular disorders and motor neuron disease, ALS. Nortina is also fully engaged and is the chair of PACTELS currently, I believe. She can correct me if I'm wrong, which is the Asia Pacific Consortium for ALS. And without further ado, I'll turn the program over to Nortina. Thank you, Nortina.
Thanks very much, Kathy. Hello, everyone. I think I should correct you, though, Kathy, that I'm not technically the chair of PACTELS. I kind of like help out with the running of PACTELS, which is the Asia Pacific ALS group. It's actually Dr. Seung Kim, who's currently the president. But anyway, it's not about me. So I have the pleasure. I wanted to just thank the International Alliance of ALS MND and specifically Kathy for inviting me to moderate this session. I'm really very excited to do this. We've got three fantastic speakers. And first of all, I'd like to introduce our first speaker, which is Professor Orla Hardiman. So Professor Hardiman is Professor of Neurology and Head of the Academic Unit of Neurology at Trinity College, Dublin. And she's also a consultant neurologist at Beaumont Hospital, where she is Director of the National ALS Service. She's a practicing neurologist for over 30 years. She also leads a research group of 50 individuals in neurodegeneration, particularly focusing on ALS-MND. She is currently the co-chair of the European Network for Cure of ALS, ENCALS, and also the European Treatment Initiative to Cure ALS, which is the TRICALS, and she's also editor-in-chief of the journal ALS-FTD. So Professor Hardiman today is going to talk to us about ALS research, time for optimism. Over to you, Professor Hardiman.
Hello, my name is Orla Hardiman. I'm Professor of Neurology in Trinity College Dublin, and I'm going to talk today about why there's grounds now for great optimism with respect to our research into ALS or motor neuron disease. We know that ALS is a complex disorder, and we know that there are probably genetic factors and that these probably interact with environmental factors that lead to the cause of the disease. Some people carry a single gene, and then other factors might happen during their life that leads them to develop the disease. In some genes it's a large number of factors, in some genes it's a small number of factors. Then there's time, and then eventually the disease develops. And our understanding of how this happens is much greater than it was in the past. What do we know about the genes? Well, there's been a significant advance in our understanding of the genetic associations with motor neuron disease, starting back with our discovery of SOD1 in 1994, and then with the evolution of technology and computational abilities, it's now possible to sequence a whole human gene and then to identify genetic variants that might be associated with the disease. And there are many different ways that this has been done over the years, as shown up here on this slide, but really about 30 genes have been identified that have a significant and credible association with the disease. But of these 30 genes, probably only 4 are really very important, particularly in populations of European extraction. Now, this might differ in populations outside of Europe because we don't have as much information about people who aren't of European extraction. It does look like that ALS or motor neuron disease is commoner in people who come from European ancestor origins. The 4 genes that are important are SOD1, TDP43, FUS, and the most important of all, which accounts for about 10% of ALS in European populations, and about half of all the familial forms of motor neuron disease, or ALS, is C9orf72. And of course, we know that that's also related to some forms of dementia. Understanding what these genes might do to lead to the disease has led to a lot of different ways that we can look at the causes of the disease, and a lot of animal work and cellular work within the laboratory has really identified quite a number of different pathways and mechanisms by which the disease can occur, ranging from problems in how we repair our DNA, through how we translate or transcribe the DNA into forming of proteins, how these proteins are removed from the nucleus, how the proteins within the cell remain stable, how our energy metabolism works within our cells, and how, if there's a damage to that energy metabolism, that can lead to disruptions, how patterns of transport of important materials up and down the nerve cable can get disrupted, and how the cellular environment, not just the neurons, but the other cells within the nervous system might be disrupted, leading to inflammation. And all of these pathways are probably important, but they may be different in different patients. So some people may have one set of pathways that are really important and that are mediated by genetic variants, and others may have other pathways. And one of the challenges that we need to understand is how these different pathways interact and how important they are in some people versus others. And it's likely that we'll be able to segregate people over time into different subgroups based on some of these different pathways. One of the ways that we've been able to do this up to now is to really model this in laboratory settings, and there's been many different ways that this has been done over the years, using mice that carry some of the human genes, particularly the SOD1 gene, to taking neurons from the mouse and growing them in cell culture, and most recently taking pieces of skin and turning the cells from the skin into neurons, and then looking to see what sort of damage might occur in those cultures from human motor neurons. And all of these different models have led to huge information, huge developments into our understanding of the disease. I suppose the most important model that we've had over the past 20 odd or almost 30 years now has been the mouse with motor neuron disease, the SOD1 mouse. And the mouse has been used extensively to test for new drugs, to see if the drug works and to see if the drug is toxic. And that's led to many drug trials. And some of the pathways that have been targeted are shown on this cartoon here, where we've had a lot of insight into how the neural nervous system within the SOD1 mouse becomes disrupted, the cells within the nervous system, and what sort of drugs that we might be able to use to interfere with that disruption and prevent the disease from progressing in the mice. And we've been pretty good at doing that. There are many drugs now that work quite well in the mouse model. The difficulty, of course, is that these drugs that have worked very well in the mouse haven't always translated into working well in the human condition. And this is what we call the valley of death, where we can be very good at looking at mechanisms and targeting new drugs within a cellular environment or even within a small animal environment like the mouse, and then bringing that into the human domain where I work, where we try these drugs in people with the disease, has been a little bit more disappointing. And all of the drugs that I showed on that cartoon have not been effective in humans, which could be perceived as being very disappointing. But what we've done there is we've learned from our mistakes. We know now that there are lots of problems in using a mouse and turning that mouse information into human information. And that's because the anatomy of the mouse differs from the anatomy of humans. And of course, mice are born and reared in a laboratory, so their genetic background is very similar, whereas in humans, the genetic background is quite different. And of course, we put lots of different gene variants into the mouse, lots of different copies of the human gene, whereas in fact, humans only have one copy of the gene, so that's the way that gene abnormality might lead to disease might be a little bit different in humans. So part of the thing that we've understood now, and part of the progress that's been made, has been our understanding of the fact that translating from mice into humans is probably not a linear thing, that we can learn lots of things about the mouse, but turning that into a treatment that works in humans is probably a little bit over optimistic, and there's a lot more work that we need to do. So we need to stop doing these studies where we're just testing the drug on mice and then translating that drug into humans and expecting the drug to work in humans. Now, we did this for a long time over the years. We kept on doing the same things. Well, maybe this drug will work in the human because it worked in the mouse. And that was what Albert Einstein defined as definition of insanity, that we kept on doing the same thing over and over again and expected to get a different result. So I think now things are moving, and we understand now the complexity of ALS or motor neuron disease in humans, and some of that complexity is a function, of course, of the fact that we're more complex brains than, for example, the brain in the fly, which is a fruit fly that we still use for modelling, or the brain in the mouse. And these animal models can give us a lot of information, a lot of insights, but we need to be, I suppose, a little bit more humble in when we translate into the complexity of the human condition. What we need to do is take a really, I suppose, a precision medicine approach towards developing new treatments. And precision medicine is a bit of a buzzword. It means that we're tailoring medical treatments to the individual characteristics of the patient or the individual characteristics of the disease in that patient. So it allows us to classify people into subgroups that then differ in their susceptibility to a particular disease and in the biological processes that underpin that disease. Of course, precision medicine has been very successful in other conditions like cancer, and what we need to do is to move that approach into the much more complex disorders that are the disorders of the human nervous system. That's because we have a very complex system of causes, where there are genetic factors and there are environmental factors. There's a passage of time, there's aging, and there are many factors here that we don't really understand very well, but that we need to study to a greater extent. What we do know, though, is that if we take a precision medicine approach, the likelihood is that we would be able to separate people into different subgroups, and these different subgroups may well require different types of treatments. And the challenge for us is to learn how to do that so that we can identify these small clusters of people with different types of motor neuron disease that we can then bring forward for different types of treatments. So our mission is to develop the right drug for the right patient in the right time at the right dose. So have we been able to do that in clinical trials? Well, of course, in the past it hasn't been very successful. We've had 40 or 50 trials that have had looked like they were very promising but turned out to be negative. We know now that this animal-based approach isn't always the best approach to take, and that we need to really think about different subgroups of patients with different types of processes that are leading to disease, and possibly different pathways that are causing the disease that we might be able to target differently. And the ways that we need to do that are to be able to identify these people using what we would call biomarkers, or markers of the different subtypes of the disease. So we need to do that, and we also need to understand the mistakes that are made in clinical trial design and outcome measures, and I think we'll be discussing this during these presentations as well. So what about the present? Well, the future is bright. The present is bright as well. there are many compounds that are in current clinical trial in phase 1 or phase 2 or some phase 3 trials, which means that the phase 3 trials are the ones that will tell us whether the drug is really working and whether the drug might come to the market in the near future. So there's a lot of interest in the pharma industry in motor neuron disease, which is wonderful, because 7 or 8 or 10 years ago that just wasn't the case. So we've many more trials coming along, and in fact some of the concerns are that there are more trials than there are patients now, which is a good place to be. The future will be to have better ways of selecting people for trials and better targeted treatments, like I said. And some of the ways that we do that, we know. Some of the ways that we can do that, we know. So, for example, we're better now at being able to predict whether somebody is going to move very slow or very fast in their disease, because my colleagues in Utrecht developed this prediction model called the NCAS prediction model, which can provide a probability of prognosis for the person. Now, obviously, that can be upsetting for people as well, but it does allow us to use a number of factors that we can discern in a clinical setting, and then to determine whether the person will be moving really slowly and therefore would be difficult to enroll in a trial, or moving at a moderate pace who would be suitable for a trial, and that would be what we would call the eligibility range for the trial. And we can do this very effectively now, and we have some trials that are going to be using this model. Now, the survival model can break groups of people into different subgroups. Some people, unfortunately, might have a very rapid progression, but some people may have a very slow progression, and the objective would be to enter people into trials based on the likely progression of their disease. And when we look at the prediction model and how it might have been used in clinical trials in the past, we can see that even though we tried to enroll people based on what we thought was the likely progression of their disease, we didn't get that we weren't very good at doing that, because we can see that many of these trials that were done, and this is work by Ruben van Eyck and Leonard Vandenberg from Utrecht, they contain patients with lots of different probabilities of disease progression, and of course that makes it very difficult to assess the trial, because we have a lot of what we would call heterogeneity or variability within the patient population. Now, if we look at how many patients we exclude in trials, and how we've been able to improve the exclusion rate, the number of people that we don't let into trials for various reasons, unfortunately we haven't been very good at doing that. So of the trials that we've engaged in over the years, we've excluded maybe 60% of people who might have benefited from the drug, from the treatment, where the treatment to work. We can do better with that with some of these new models of prediction that we have. So on average, up to in the previous trials that we've done, 60% of people were excluded before we even got to enroll them into trials. And unfortunately, over the years, there wasn't really an improvement in that, even though we tried very hard to do that based on looking at previous trials and see if we could refine the criteria for entry into clinical trials. There's a great deal of variability across the trials. Of course, for example, the edaravone trial, which led to approval in the US, excluded about 90% of patients. And by contrast, one of the cytokinetic studies, the Tirzenta trial, excluded about 50% of patients. So there's a huge variability in the percentage of people that we exclude in trials. Of course, we prefer to exclude nobody. That is difficult if somebody has a very slowly progressive form of the disease. The way we measure disease progression at the moment makes it difficult to enroll these people. Now, the trials that we're currently enrolling in Europe, I've listed some of the major ones that are in progress at the moment. And of these, the Lighthouse trial, which is using an anti-AIDS drug, and the Magnet study, which is a new study being developed within the TRICALS group, both use this TRICALS prediction model rather than the old-fashioned way of enrolling people. So that's a really exciting development. There are also new designer treatments based on genetics, and that's really exciting as well. And there have been a number of studies now. The tofersen study that's targeted at SOD1. And there's a number of studies now also targeting the gene C9orf72. So this is true precision medicine. This is based on people who have the genetic variant, and we put in a treatment that turns off the gene, and hopefully that'll slow the progression of the disease. So that's a really exciting and important innovation and development. We'll make a lot more progress, of course, if we all work together, because ALS is a rare disease, and we need to understand the disease in all of its iterations, from genetic to the clinical progression. From a genetic point of view, of course, we've all been working together now for a number of years in the Project Mind consortium, and that's a way of looking at the genetic variants that are associated with ALS or motor neuron disease. And there are many, many countries involved in the Project Mind consortium, where we take a sample of DNA from patients, we put it into the Project Mind consortium, and each country then funds the analysis of the DNA from the patients within their own country, and that's led to many, many new insights into our understanding of the causes of the likely cause of ALS. And Project Mind is moving apace, and there'll be many presentations at the symposium on the new insights from Project Mind. We need to also capitalise on the way that we developed Project Mind. So Project Mind was a transnational collaboration involving 17 countries. We want to expand that to a global collaboration, and then to translate these genetics into clinical studies. And then using the Project Mind mentality, idea of cross-national collaborations, we want to develop these collaborations to understand aspects of the disease that aren't just genetic, but the clinical aspects of the disease and the environmental factors that may lead to disease development. And of course, that's what we do in TRICALS, the Treatment Initiative to Cure ALS, which is a group of 44 centres across Europe who are interested in clinical trials for people with ALS and interested in improving clinical trials and developing this new highway to a cure for motor neuron disease or ALS across Europe. And within TRICALS, we've developed 5 lanes of research that in which we want to engage, including really developing information at scale by combining our different data sets, by developing new biomarkers, by improving clinical trial design, by putting this platform together where we can collect all the information in real time and interrogate it in real time, and of course to be alive and sensitive to the needs of people with the disease, and not just to move ahead with what we think is best, but to incorporate the views of the people who have the disease as well. So TRICALS has OIPALS as a member, and and a representative from OIPALS, Evie Revier, is on the board of TRICALS, an executive group of TRICALS. We want to be better at collecting our data, and we're going to develop this pan-European electronic patient platform across the different sites in Europe. This is what it looks like. So we'll be collecting information from people with ALS, and then collecting as much as we can about those patients on this patient data platform, so that we can then start undertaking very detailed analysis at a very large scale. And I'm pleased to say that this is actually funded through a programme within Ireland, in collaboration with colleagues within the TRICALS group, and with some funding sources provided through the Irish Exchequer. It's very important, like I said, that we recognise the needs and values of people who have the disease, so that we understand the experience of the illness beyond what's actually happening at a pathological level, but what's happening to the person and their family as they experience the disease. So a really important part of what we do is to have the patients and the families as collaborative partners in these projects, and to really understand the meaningfulness of the outcomes that we measure, and also to incorporate outcomes that are important to the patients and their families, using these new technologies that we've been developing, of course, as a response to COVID as much as anything else, but to reduce the burden of collecting information on people when we're doing this in our clinical practice, but also in the context of clinical trials. So to conclude, we know that ALS is a complex condition, that people with ALS have different subtypes. That's really important insight. It's something that we hadn't really incorporated into our understanding of the disease, and how we manage disease, and how we think about developing new treatments for the disease. So recognising that is very important. The complexity of the disease requires that we work together, and that'll help to speed things up in terms of new drug development. And then there are definitely new and more effective treatments on the horizon, and I think there is grounds now to be really optimistic about the work that's happening today, so that tomorrow I think that we'll have better and more effective treatments.
Right. Thank you very much, Professor Hardiman, for that talk and telling us all about the state of the art with respect to developing new treatments.